Okay, so welcome back to this next video in which we are discussing Cys-Loop receptors. Okay, right, so we're in the process of discussing the extracellular domain of Cys-Loop receptors. Okay, so here is the N-terminus of the Cys-Loop receptor subunit, and here is the portion that then goes on to become the M1 helix. And we've seen that the extracellular domain, which is all of this portion between the amino terminus and the beginning of the M1 helix, which is the start of the transmembrane domain, consists of this beta sandwich, these two beta sheets that are opposed to one another. Okay, and we've seen the complicated way in which the polypeptide builds this beta sandwich structure here. Okay, so this means that now there are two sides to the extracellular domain. Okay, there is this positive side, which is this outer beta sheet here, okay, which is the side that will be on this side of the subunit here. Okay, and then we've got the negative side, which is this inner sheet here, which will be on this side over here. Okay, and these sides of these sheets will be the portions facing into the pore, or the cavity, the central cavity made by the 5 extracellular domains of the 5 Cys-Loop subunits. Okay, and this side will be the sides facing outwards. Okay, right. We've also discussed the fact that ligand binding domains on Cys-Loop receptor pentamers are in between neighbouring extracellular domains of neighbouring subunits, basically. Okay, and the binding sites are going to be made up by the loops A, B, and C from the positive face of one of these subunits, and loop D, which isn't a loop at all but part of a beta strand, but is called a loop, loop E and loop F from the negative part of the adjacent subunit, basically. Okay, and that together will make the ligand binding site. These will provide the residues in to make the ligand binding site. Now note, what I have shown you is the basic structural features that are conserved for all Cys-Loop ligand-gated ion channels. Okay, however, again, I will stress the fact that the primary sequence of amino acids in these different Cys-Loop ligand-gated ion channel subunits are going to be very different, and that is why different Cys-Loop receptors are capable of binding to different ligands, because these loops, although they're conserved in the different Cys-Loop ligand-gated ion channels, they will have different amino acids actually in them, and therefore you'll be able to bind different ligands in this interface between adjacent subunits. Okay, right, so what does this overall mean? Well, this now means, and in fact I'll show it on this picture up here, okay, this now means that in principle every single Cys-Loop ligand-gated ion channel pentamer has 5 binding sites, one between each of these, one at each of these interfaces between adjacent subunits, basically. Okay, and there are 5 interfaces, therefore we would expect there to be 5 binding sites. Now indeed, in some Cys-Loop ligand-gated ion channels, there are actually 5 binding sites. Okay, these are generally in the homopentamers. Okay, so we now have some important terminology to come up with. Okay, so homopentamer, what is meant by a homopentamer? A homopentameric Cys-Loop ligand-gated ion channel is one where the subunits that you use to build your Cys-Loop ligand-gated ion channel pentamer are all identical. Okay, so for instance, gluCl, enic, and glic are all homopentamers because the subunits that we use to build those were all identical. However, in the mammalian grape families, nicotinic acetylcholine receptor family, the GABA A receptor family, the glycine receptor family, the 5-HT3 receptor family, there's going to be absolutely loads of subunits that are all different, which you can use to build those types of ion channel. Okay, and you don't necessarily have to make homopentamers at all. Okay, so in homopentameric ones, where the 5 subunits that make the cis-loop ligand-gated ion channel are identical, then of course you will have 5 ligand binding domains, because how could you not? If you have one, then all the others must be identical to it, so they must also bind ligand. Okay, simply by the symmetry of it, you can either have none or you can have 5. You can't have anything in between that. Okay, however, the important concept here is that many of the important receptors that we are going to see are not going to have 5 ligand binding sites, and the reason for this is that they're not homopentamers, they are instead heteropentamers. Okay, so to get this concept across, what I'm going to show you is the structure of the nicotinic acetylcholine receptor, which is found in skeletal muscle cells in humans and other mammals, basically. Okay, so I'm going to show you the muscle type nicotinic acetylcholine receptor, okay, which is a heteropentamer, basically, okay, which means the subunits that you use to build the pentamer are not the same, basically, and therefore you don't have to have that symmetry. So although you will, in principle, have 5 binding sites, it's not necessary that these binding sites can actually bind the ligand in all cases. Okay, and therefore it's not necessary that all of the binding sites are actually functional and will bind the ligand for that receptor. Okay, so let me show you the muscle type nicotinic acetylcholine receptor. And I'll come down here to show it, but it's a little bit squashed up there. In fact, actually no, I will sort of squeeze it in here. Okay, so we're viewing from the extracellular side, basically. Okay, and I'm going to show you the 5 subunits that are used to build this nicotinic acetylcholine receptor. Now, this might not make sense initially when you see it, but we will go over the nicotinic acetylcholine receptor family in much more detail later. For now, what you need to understand is that the subunits out of which you build CysLoop ligand-gated ion channel nicotinic acetylcholine receptors, there are loads of them. There's not just one of them. It would be beautifully simple if there was just one subunit for a nicotinic acetylcholine receptor, and we could call that the nicotinic acetylcholine receptor subunit, like we could with gluCl and elic and glic, and then all you'd have to do is assemble a homopentamer of these, and then you'd have your nicotinic acetylcholine receptor. It's not the case. There are absolutely loads of different subunits that you can use to build nicotinic acetylcholine receptors. Okay, and therefore you can combine different ones together to make heteropentameric nicotinic acetylcholine receptors, where the subunits that make up the pentamer are not all identical. Okay, so I'm now going to show you this muscle-type nicotinic acetylcholine receptor, which is going to contain 4 different types of nicotinic acetylcholine receptor subunit. And as I say, later on we will go through all the different types of nicotinic acetylcholine receptor subunit. Okay, so that will come up later. Okay, but for now I'm just trying to illustrate the point that there isn't always actually 5 functional ligand binding domains. Okay, so here is my first subunit that I'm going to show, which is called the alpha-1 subunit of a nicotinic acetylcholine receptor. Then after this, you'll have either gamma or epsilon, depending on whether we're talking about the fetal muscle-type nicotinic acetylcholine receptor or the adult one. Okay, so this is an important point. If you are a fetus, you will use the gamma nicotinic acetylcholine receptor subunit here, okay, and if you're an adult, you will use the epsilon nicotinic acetylcholine receptor subunit here. Okay, so the gamma nicotinic acetylcholine receptor subunit is used in the fetal muscle type nicotinic acetylcholine receptor. The epsilon nicotinic acetylcholine receptor subunit is used in the adult muscle type nicotinic acetylcholine receptor. Okay, so I'll just sort of box that in orange here. Okay, right. Then after the gamma or the epsilon subunit, what you then have is another alpha-1 subunit here. Okay, so you have 2 alpha-1 subunits of nicotinic acetylcholine receptors. Then you have a delta-1, and then finally a beta-1 subunit of nicotinic acetylcholine receptors. Okay, so for now, these are all just different proteins, basically. Okay, so alpha-1 is different from beta-1, it's different from delta, it's different from gamma and epsilon. They're all slightly different proteins. They will have different primary sequences of amino acids. They'll all have the basic core structures that I have spent so long talking about. They'll all have extracellular domains that roughly look like this. They'll all have transmembrane domains that look like the things that I've shown you, and intracellular loops that look like the things that I've shown you, but they will all have fundamentally different sequences of amino acids, and therefore they are different proteins. Okay, so although you have 5 interfaces here where potentially acetylcholine could bind, in reality not all of those interfaces actually do bind acetylcholine. After all, they do all have different sequences of amino acids, and therefore they will have different sequences of amino acids in their loops A, B, C, and D, E, F, basically. Okay, and therefore these binding sites might be able to bind acetylcholine, or they might not. In reality, you only have 2 acetylcholine binding sites here, and they're on the positive sides of the alpha-1 subunit. So here is one of them, and that's using the positive side of the alpha-1. Okay, so remember, this is the positive face of alpha-1, so that will contain loop A, loop B, loop C, and it uses the negative side of the gamma or the epsilon subunit, which will have loop D, loop E, and loop F making up that binding site. Okay, so that's actually capable of binding acetylcholine. The binding site here isn't, basically. Okay, so the positive side of the gamma and epsilon subunit, or epsilon subunit rather, and the negative side of the alpha-1 subunit is not a valid acetylcholine binding site. It will form a little binding site there, but it's not for acetylcholine, basically. Okay, the other acetylcholine binding site that's actually functional here is the one between alpha-1 and delta, and again that involves the positive aspect of this alpha-1 subunit and then the negative aspect of our delta subunit there. Okay, right, so in this muscle-type nicotinic acetylcholine receptor then, you only have 2 actual binding sites for acetylcholine, even though you do have these 5 interfaces, basically. Okay, so that's an illustration of why it isn't always necessary that cis-loop ligand-gated ion channels have 5 binding sites for the ligand, because some of these interfaces aren't actually going to be able to bind the ligand at all, basically. Okay, right. So now what I want to talk about then is I want to talk about the actual gating mechanism. I want to talk about how ligand binds and what is understood about how this then actually breaks the hydrophobic girdle and therefore opens the channel. Okay, so firstly I'm going to discuss ligand binding. How does ligand actually bind to these interfaces? We've discussed that loop A, B, C on the positive interface of one of the subunits will be part of the ligand binding pocket, and loop D, E, and F on the negative face of the other adjacent subunit will be part of the ligand binding pocket. But we now want to discuss how does the ligand actually bind. Well, this is going to be different in the case of different cis-loop ligand-gated ion channels, because after all of the different ligands have different properties. So what I'm going to discuss is what is known about how acetylcholine binds to the ligand binding pockets of nicotinic acetylcholine receptors. Okay, but this isn't going to generalize, basically. This is specific to acetylcholine binding to the nicotinic acetylcholine receptors. Okay, but I'm going to give it just to illustrate the point that ligand will bind in some way. Okay, right. So, how then does acetylcholine bind to these binding pockets on nicotinic acetylcholine receptors? How does, what special features do these loops that make up the binding site of an acetylcholine receptor actually have in order to bind the acetylcholine? Well, basically, the way that acetylcholine binds to these binding pockets is going to make use of the fact that acetylcholine has a positive charge. Okay, so if I bring back my picture in which we have acetylcholine, Okay, here is my picture of acetylcholine. Remember, because of this quaternary ammonium group here, the acetylcholine has a positive charge. Again, this positive charge is going to be key to the way that the acetylcholine can bind in these ligand binding pockets on the nicotinic acetylcholine receptor. Okay, so basically, in these binding pockets here, and we'll use this muscle-type nicotinic acetylcholine receptor as an example, but there are many other types of nicotinic acetylcholine receptor, as we'll see later. So basically, in this binding pocket here, we have loop A, B, and C being contributed to by the alpha-1 subunit, and I think I'll just put this down here. Okay, so I'm going to draw out those two subunits again. So here is the gamma and epsilon subunit here, and here is the alpha-1 subunit here. Okay, and basically they're forming a binding pocket here, and the way people often show this is they draw three little loops here to illustrate the loop A, loop B, and loop C, and for some reason they usually put loop A on the outside even though it's actually on the inside. Okay, so loop A, loop B, loop C. Okay, and then we'll have the three loops on the other side, which will be loop D, loop E, loop F, and again people generally just schematically put D on the outside, E in the middle, and F on the inside, like so. Okay, and basically these loops are then going to contribute residues that are going to assist in the binding of acetylcholine. So what sort of interactions then underlie the binding of acetylcholine to residues within these loops? Well, basically they're going to be cation pi interactions. Okay, these loops that make up the ligand binding pocket for an acetylcholine molecule generally have a lot of big, bulky aromatic residues within them. Okay, so let me give you examples of big, bulky aromatic residues. So tryptophan is one. Okay, another example is tyrosine. So let me just draw the R groups of these amino acids out. So we've seen the R group of tryptophan when we were discussing the structure of 5-hydroxytryptamine, but I'll just draw it again. Okay, so I won't draw the core amino acid structure, I'm just showing the R group of the amino acid now. So remember, tryptophan consisted of a methylene group in its R group. Okay, then it had this pyrrole ring, okay, which remember had the nitrogen at the tip here, okay, and then 4 carbons making up the rest of the ring here. And you have a double bond there, and a double bond there. Okay, whoops. And then that nitrogen will have a hydrogen attached to it here. Okay, right. And then to turn the pyrrole ring into an indole ring, what you then have to do is attach a benzene ring onto the pyrrole ring here. So here is our benzene ring. So that's the R group of tryptophan. Now clearly that's very big, so it's a bulky residue. Okay, in addition it's very much so aromatic because it contains these alternating double and single bonds which will create delocalized rings of electrons basically. Okay, and we'll discuss delocalized rings of electrons in a bit more detail in a moment. Another example of a bulky aromatic residue is tyrosine. Okay, and tyrosine, its R group consists of a methylene group, then followed by a benzene ring, so here's the big bulky aromatic group, okay, like so, and then you have an alcohol group coming off that benzene ring. Okay, and basically these loops will contain tryptophan and tyrosine residues in the case of these binding pockets for nicotinic acetylcholine receptors, which are intended for acetylcholine. Okay, now how does this help in binding to acetylcholine? Well, to understand that, we need to discuss the delocalized ring of electrons that you have above and below benzene rings in a bit more detail. So let me just remind you of a bit of basic chemistry here. So basically, when you actually produce these benzene rings, okay, it turns out that their structure isn't actually really alternating double and single bonds. We show it like that, okay, but in reality it's not alternating double and single bonds. If it was alternating double and single bonds, then benzene rings would not be perfect hexagons because double bonds are actually shorter than single bonds, so it would not make a regular hexagon if you had alternating double and single bonds. However, benzene is a perfect regular hexagon. Okay, so really the way you should think of benzene is every single one of these bonds is like 1.5 bond, basically, so they're all equivalent. And what really happens is you have this ring of carbons here, okay, and all the carbons are singly bound to one another. And then these 6 electrons that will be in these second bonds here, okay, those go into delocalized rings that lie above and below the plane of the ring of carbon atoms. So here's one of these delocalized rings that I'm now showing, which is above the plane of the ring of carbon atoms, and now I have the one that's below here. So here now is another ring of delocalized electrons that is below the plane of the ring of carbon atoms. Okay, so basically in the structure of benzene, 6 electrons end up in these delocalized rings. And these delocalized rings then end up with a negative charge. Okay, so they are centers of negative charge, and the ring of carbons here ends up with a sort of positive charge. Okay, now why is this ideal for binding acetylcholine? Well, basically, if these loops are providing in lots of these aromatic residues, these bulky aromatic residues, then the idea is we have lots of these delocalized rings. Okay, and there's a special piece of terminology here. People refer to the electrons in these delocalized rings as pi electrons. Okay, so that's an important thing to know. And basically, what can now happen is we're going to have lots of these delocalized rings with these pi electrons, which are forming these centers of negative charge. And the idea is that what can now happen is the acetylcholine can go in there. And acetylcholine has a positive charge. Okay, so the idea is that the positive charge of acetylcholine, and I'll just represent acetylcholine as ACH here. Okay, the positive charge of acetylcholine can interact with the negative charge of one of these delocalized rings of electrons, okay, and that can form an electrostatic interaction between the acetylcholine and the binding pocket, basically, and it's thought that that is how acetylcholine binds in these binding pockets of nicotinic acetylcholine receptors. By these interactions between the positive charge on the acetylcholine molecule and these centers of negative charge, which are the pi electrons in these delocalized rings associated with tryptophan residues and tyrosine residues. Okay, right. So there is a special name for this sort of a bond. It's known as a cation pi bond, basically. So when you have a cationic molecule like acetylcholine binding to pi electrons of delocalized rings, that's called a cation pi interaction. So we believe that the way that acetylcholine binds in these acetylcholine binding pockets between adjacent subunits of nicotinic acetylcholine receptors is through cation pi interactions. Okay, now, as I say, that is completely specific for the nicotinic acetylcholine receptors. It doesn't then generalize to the other CysLoop ligand-gated ion channels. Okay, what does now generalize to other CysLoop ligand-gated ion channels is what happens after the ligand has bound in this ligand binding pocket. Okay, what's believed to happen once ligand has actually bound to a CysLoop ligand-gated ion channel is that the extracellular domains are going to end up rotating, and this rotation is going to be carried on to the M2 helix, basically, and then it's going to end up disturbing the hydrophobic girdle. Okay, so let me go over the page and we'll discuss this a little bit more. Okay, so firstly let me draw out again a CysLoop ligand-gated ion channel where we are viewing it from above and we're seeing the 5 extracellular domains. Okay, so here is that central cavity once again. Here are 5 extracellular domains here, like so. Okay, and we will take as our example once again this muscle-specific nicotinic acetylcholine receptor. Okay, because it will highlight an important principle. Okay. Which is what happens in these receptors where you only have 2 ligand binding domains rather than all 5, or rather more generally where you have less ligand binding domains than 5. Okay, so here is the alpha 1, here is the gamma or the epsilon subunit, here is another alpha 1 subunit, here is delta, and here is beta 1. Okay, right, and we know that the place where the ligands bind is there's one ligand binding site there between alpha 1 and gamma or epsilon, and there's one ligand binding site there between alpha 1 and delta. Okay, so what actually is believed to happen then when ligand binds, and this generalizes to the other CysLoop receptors as well. Okay, so we've got a specific example here to illustrate the point, but it's true for the others as well. Okay, so when ligand binds, what's believed to then follow is you get a rotation of the extracellular domain of the subunits of the ligand-gated ion channel, okay, and it's a rotation in the clockwise direction. So let me show you this. Okay, so what's believed to occur then is that the alpha 1 subunits will rotate round in a clockwise direction when the ligand actually binds in these ligand binding pockets, okay, and they'll rotate around by around 15 degrees. And basically, even the ones which are nowhere near these ligand binding pockets, such as the beta 1 subunit, it's believed that when these other ones rotate round clockwise, the other one has no choice really but to rotate round with it. It's like they're all cogs, and when one rotates, the others have to rotate as well. So beta 1, even though it's nowhere near a ligand binding domain, it's going to rotate around as well. So the thought is that all of the extracellular domains of all of the 5 subunits of the CysLoop ligand-gated ion channel are going to end up rotating in this clockwise direction by around 15 degrees when ligand actually binds. Okay, and the reason I wanted to show this for this muscle-specific nicotinic acetylcholine receptor is because this highlights that this still works even when you don't have 5 ligand binding sites. Okay, it might be perfectly obvious when you do have 5 ligand binding sites that every single one of them will rotate. Okay, but when you don't have 5 ligand binding sites, and when you have subunits that don't actually have contact with a ligand binding site, it's not totally obvious that all of them will then rotate. But this illustrates the principle that it's like cogs basically. When one rotates, all of the others have to rotate as well. So they all rotate in unison basically, in this clockwise direction, by around 15 degrees. Okay, when the ligand actually binds in those ligand binding domains. Okay, and this, as I say, generalizes for all the different CysLoop ligand-gated ion channels. So when ligand binds in the ligand binding pockets of the CysLoop ligand-gated ion channels, no matter how many binding pockets you have, all of the extracellular domains of those CysLoop ligand-gated ion channel subunits are going to start rotating in the clockwise direction by around 15 degrees. Okay? Now, that's just the extracellular domain so far rotating. How does this get transmitted to the M2 helix, which is what we need a conformational change to occur in, because that's, remember, where the hydrophobic girdle is, and somehow we need to break the hydrophobic girdle. Okay? And indeed, what's going to happen is this rotation is going to be transmitted to the M2 helix, and it's transmitted through portions that contact the M2 helix. Now, the 2 portions which contact the M2 helix of the extracellular domain are this beta 1, beta 2 loop here, okay, and also the cis loop. So let me just underline these. So the beta 1, beta 2 loop contacts the M2 helix, and the cis loop contacts the M2 helix. Okay, and basically when this rotates in the clockwise direction, and it will be rotating round like that, okay, because this is the front when we're viewing from this sort of side here, and this is the back, and we're going to be rotating round like this, so it's going to be going round like this. Okay, what will then happen is the beta 2 and beta 3 loop and cis loop, which contact the M2 helix, they cause the same rotation in the M2 helix basically of the transmembrane domain. And what this then does is it disturbs that hydrophobic girdle. So if I get the picture back of the hydrophobic girdle here, okay, it disturbs the interactions between those hydrophobic residues in that ring. Okay, so when the M2 helices start rotating, like so, in this clockwise direction, Okay, it disturbs the interactions between the hydrophobic residues. The hydrophobic girdle gets broken, therefore, and then what happens is the M2 helices spring back outwards, opening up that pore. Okay, and then you've opened the gate now. Ions will move through. Okay, right. So that then now is the gating mechanism by which ligand binding to the extracellular domain of the CysLoop ligand-gated ion channel can actually lead to the hydrophobic girdle being disturbed and therefore the opening of the actual pore down the middle of the transmembrane domain. Okay, and as I say, the only part of that doesn't generalise to all of the CysLoop ligand-gated ion channels was the portion about the cation pi interactions being really important in the ligand binding pockets of nicotinic acetylcholine receptors. Okay, that only applies for the nicotinic acetylcholine receptor family within the CysLoop ligand-gated ion channel family. The twisting of the extracellular domains, that applies for all different CysLoop ligand-gated ion channels. Okay, right. So now what we're going to do is we're going to now begin a more in-detailed study of all of the different examples of CysLoop ligand-gated ion channels, starting with the nicotinic acetylcholine receptors.